everyone, in this video we are going to learn in detail about the process of microsporogenesis. In previous video we have discussed about the transverse section of anther, so let's quickly revise that once. We all know that in flowering plants, a flower has a male reproductive structure which is called stamen. Okay, the stamen is having two parts, filament, and on the top of this there is a lobe-like structure present which is called anther. Okay, now if you take the transverse section of this anther, that means if you will cut this anther along its width, okay, if you'll cut this anther along its width and observe this thin slice of anther under microscope, you can find out there are different layers of cells present in the anther. Okay, now the outermost layer of cell which is present around this anther is known as epidermis. Okay, here epi means outer, and the meaning of this term dermis, this term dermis comes from another term called derm. Okay, so the meaning of this term derm is skin. Okay, so epidermis, and epidermis can be found as the outer layer of cells in all the organs of the plant, in root, leaves, branches, thin stems or leaf stalk. Everywhere epidermis is actually present as the outermost layer of the cell. This is actually protective in function. Okay, next to epidermis we have another layer of cell which is called endothecium. So here the meaning of this term endo is inside. Inside means the layer of cell which is present inside to the epidermis. After endothecium we have 3 to 4 layer of cells, and these layer of cells forms middle layers. Okay, and then next to middle layer we have another layer of cell called tapetum. Okay, now inside tapetum there is actually a cavity present, and this cavity is known as microsporangium. Okay, as we have already discussed in the previous videos, we know that the anther of most of the angiospermic plants is dithecous. Okay, and inside this dithecous anther there are cavities present, and these cavity actually forms 4 microsporangia. Okay, so a dithecous anther always have 4 microsporangia. This term microsporangium is actually singular, and microsporangia means plural. Okay, and this microsporangia is actually filled with the cells, and these cells together form sporogenous tissue. Okay, so you have microsporangia or microsporangium, and these microsporangiums are actually filled with the tightly packed, or you can say similar type of cells, and these cells together form sporogenous tissue. Okay, so initially when the anther is young, at that time the size of this microsporangium is obviously small, because the anther is very young and it is still growing in size, and it is still in the developmental phase. Okay, so at this time the microsporangium is smaller in size, and all the cells, they are tightly packed inside the microsporangium. Okay, all the cells of sporogenous tissue. Okay, now remember here, all these cells of sporogenous tissue, they are actually capable of producing microspore or pollen grains. Okay, they are capable of producing microspore or pollen grains. Okay, some of these cells of sporogenous tissue, they actually degenerate, and their degenerative mass is absorbed by the other cells present inside the microsporangium. Okay, and in the same time the anther is also growing, and anther is also developing. Hence the size of this microsporangium now increases. Okay, and the cells of this sporogenous tissue are now present inside the microsporangium with little space between them. Okay, with little space between them. Okay, so remember here that there is no increase in the size of the cells. Okay, there is no increase in the size of the cells, only there is increase in the size of the microsporangia. Okay, due to which the space between these cells increases. Okay, and all these cells, as they are capable of producing microspores or pollen grains, they are known as microspore mother cells or pollen mother cells. Okay, microspore mother cells or pollen mother cells. And in the initial phases, all these cells are diploid. That means they have 2 sets of chromosomes. Okay. Now what happens is this microspore mother cell undergoes meiosis. Okay. It undergoes meiosis. It undergoes meiosis 1 and meiosis 2. Okay. So this microspore mother cells or pollen mother cells, it undergoes meiotic cell divisions. Why meiosis? Because meiosis is actually a type of cell division which is also known as reductional division. As in this, the number of chromosomes present in the parent cell are reduced to half in the daughter cells. Okay, the number of chromosomes present in the parent cell are reduced to half in the daughter cells. For example, if there are 36 cells, for example, if there are 36 chromosomes in the in this particular microspore mother cell, okay, and now this is formed, this pollen grain or microspores are formed. So inside this pollen grain or microspore, how many chromosomes will be present? 18 chromosomes each. Okay, 18 chromosomes each. Okay, so this type of cell division in which the chromosome number is reduced to half is known as meiosis or reductional division. Okay, meiotic cell division or reductional cell division. So complete set of chromosome is represented by 2n and half set of chromosome is represented by n. Okay, so these microspores or pollen grains which are formed here, they have only half set of chromosome. So this process in which the microspore mother cells or pollen mother cells present inside the microsporangia of anther undergoes meiotic type of cell division and produces haploid microspores or pollen grains. Okay, so this process is known as microsporogenesis. Okay, so remember here that microsporogenesis is the process in which the microspore mother cell or pollen mother cell present inside the microsporangia of anther undergoes meiotic type of cell division and they produce haploid microspores or pollen grains. Okay, they will produce haploid microspore or pollen grains. Okay, so this process is known as microsporogenesis. And one question which is asked in the exam is, where does the process of microsporogenesis takes place? Okay, or what is the site for the process of microsporogenesis? So it occurs inside the microsporangium of anther or in a microsporangium of anther of the stamen or male reproductive part of the flowering plant. Okay, and another point to be noted here is when this microspore mother cell, this is one microspore mother cell, okay, it divides and produces four microspores. Okay, and all these four microspores, they are not free. Okay, they are not simply free to move here and there inside the microsporangia. These microspores, four microspores together, are actually held with each other. Okay, they are held together with each other with the help of a polysaccharide, and this polysaccharide is actually known as callose. Okay, so these microspores, when they are formed initially from the microspore mother cell, they are not free to move here and there. They are held together, or they are present in the colony of 4. Okay, they are present in the colony of 4. Okay, they are present in the number of 4, and all these 4 microspores, they are held together by a polysaccharide which is called callose. Okay, and these microspores, they actually form microspore tetrad. Okay, what they have formed? They have formed microspore tetrad. Okay, a tetrad means, here the meaning of this term tetra is 4. So 4 microspores or pollen grains now are held together with each other with the help of a polysaccharide called callose. Now there are different types of microspore tetrad, and this particular topic is important for your NEET exams. Okay, there are different types of microspore tetrad. The first type of tetrad is called tetrahedral tetrad. This is the most common type of tetrad present in most of the dicot or most of the angiosperms or angiospermic plants. Okay, in this type of microspore tetrad, what happens is 3 microspores you can see from one particular plane. Only 3 microspores are visible, okay? And the fourth microspore is actually present on the back side or on the another side, on the another plane, okay? Then next type of tetrad is isobilateral tetrad, okay? In this type of tetrad, all 4 microspores can be viewed or can be seen from one particular plane, okay? All 4 types of microspores can be seen from one particular plane, okay? And in this all these 4 microspores, they are actually arranged on the same side or on the same plane, okay? The third type of microspore tetrad is actually decussate type, in which 2 plus 2 microspores are actually arranged perpendicular to each other, okay? These 2 microspores are actually arranged perpendicular or at 90-degree angle to the another 2 microspores. This is one microspore, and the second microspore can be seen or observed on the other side, okay? Other side of this particular tetrad, okay? Next microspore tetrad is T-shaped tetrad, okay? In this actually the 2 microspores are arranged in transverse plane and the 2 microspores are arranged in longitudinal plane. So you can see here on the top 2 microspores, these 2, they are arranged in the longitudinal plane, that means along the length like this, and the rest 2 microspores, they are arranged in the transverse form, okay, or in the transverse plane, longitudinal plane and in the transverse plane. The last type of microspore tetrad is linear type of tetrad, and in this all the 4 microspores, they are arranged next to each other, okay? So they are arranged next to each other in the linear fashion or in the linear manner, okay? So quickly recap this once. Remember, this is actually the cells of the tapetum layer, okay? Inside cells of the tapetum layer, microspore mother cell or pollen mother cells are present. These microspore or pollen mother cells, they undergo meiotic type of cell divisions. In meiotic type of cell divisions, what happens is, in meiotic type of cell division, the chromosome numbers are reduced to half, okay? If the pollen mother cell is having 36 chromosomes, so the microspores or the pollen grains, they will have only 18 chromosomes. The pollen grains or microspores, they have only 18 chromosomes. That means in meiotic type of cell division, the number of chromosomes are reduced to half, okay? And initially, these microspores or pollen grains, they are not free to move here and there, okay? These microspores or pollen grains in the initial stages, they are actually held together, or they are actually binded with each other with the help of a polysaccharide which is called callose, okay? Now the cells of this tapetum, okay, very important point, note here, the cells of this tapetum secretes an enzyme which is called callase. This callase will actually digest the polysaccharide called callose, okay? Now once this callase completely digests the polysaccharide called callose, all these microspores or all these pollen grains, they are completely free from each other, okay? Now they stay as an individual structure inside the microsporangia, okay? So now these microspores or pollen grains are formed, okay? Earlier they were held together by callose. Callase is the enzyme which is secreted by the cells of tapetum. This enzyme digests this callose, and now microspores or these pollen grains are completely free from each other and they are present as an individual inside the microsporangia, okay? But even now when this microspore or pollen grain is formed, even now this pollen grain is not yet ready to be released from the anther for the process of fertilization, okay? Still this pollen grain has to undergo certain changes or certain development, okay? Right now the pollen grain is actually having a plasma membrane. You can say this pollen grain or this microspore, okay? So this pollen grain or microspore is actually having a plasma membrane. Inside this plasma membrane, we have cytoplasm. So this pollen grain is filled with cytoplasm. plus there is a haploid nucleus. Okay. So at this particular stage, this pollen grain is not actually, ready to be released in the outer surrounding. Okay. In the surrounding for the process of pollination. This pollen grain has to undergo certain changes. Okay. It has to undergo certain development. Okay. So this pollen grain actually has to develop into male gametophyte by undergoing certain changes. Okay. So in next video, we will understand and we will learn about how this pollen grain develops into the male gametophyte. Take care till then. Bye-bye.